Hello, what's going on, Gracians? Welcome back to another episode of The Scientific Mind with Sir Eman. For today's discussion, we will tackle about one of the topic of genetics, which is all about the non-Mendelian pattern of inheritance. To start our day, let's have our prayer. But before we proceed with our discussion today, I will discuss first what you are going to accomplish this week. So this is the worksheet that you are going to pass or submit to me at the end of this week. After watching the part 1 of that video, you are going to answer this activity entitled Phenotypes and Genotypes in Incomplete Dominance. So this is the worksheet. Procedure: you are going to show the possible outcome of the cross between the problem 1 using the Punnett square. After doing the two Punnett square, for problem number 1 and 2, you are going to answer these 4 guide questions. On the second day, you are going to answer the activity entitled Mystery Bull. So before answering this worksheet, read the problem first, then answer the Punnett square. For the first Punnett square, you are going to cross this 1. For Punnett square 2, you are going to use this one. And for the last Punnett square, you are going to use this one. After answering the Punnett square and showing the possible outcomes, you are going to answer the 5 guide questions. On the third day, you are going to watch the part 2 of that video. Then on the fourth day, you are going to answer the activity, What's your blood type? There are 2 problems in this activity. Activity A, you are going to determine the possible blood type of the father based on the blood type of the mother and the child. Next, for problem B, you are going to show the possible outcome that can be found in each offspring and write the blood type for each offspring. So you are going to complete this table. After doing the first and the second problem, you are going to answer guide question 1 to 3. You don't have to watch the video for one day because this video is divided into 2 parts. Part 1 and the part 2. For the part 1, you are going to watch it on the first day, then answer the activity Phenotypes and Genotypes in Incomplete Dominance. Then the second day, you are going to answer the activity Mystery Bull. On the third day, you are going to watch the second part of this video, then answer the activity What's your blood type on the fourth day. Okay, so without wasting any more time, let's start the discussion. This chapter about heredity, inheritance, and variation is divided into 2 parts. The first part, which involves the non-Mendelian pattern of inheritance, and part 2, the multiple alleles. Now let's begin this discussion about the non-Mendelian pattern of inheritance. But before that, let's discuss where these two topics is mainly rooted. The genetics. So what is genetics? Genetics is the branch of biology that studies hereditary information. It deals with the mechanisms of heredity. What is heredity? Is heredity different from trait, or is heredity similar to genes? Heredity is the passing of traits from parents to the offspring. Example of these characteristics includes eye color. Eye color can be brown or black or hazelnut. Height can be tall or short. Texture of hair, which can be wavy, curly, or straight. Shape of earlobes, which can be attached or broad. And skin color can be fair or dark. Gregor Mendel. Gregor Johann Mendel is known as the father of modern genetics. Born in Austria in 1822, he discovered the basic principles of heredity through experiments in his monastery's garden. Mendel's observation became the foundation of modern genetics and the study of heredity, and he is widely considered as a pioneer in the field of genetics. In his observation on pea plants, he found out that the seed of the pea plant can be round or wrinkled. Its color can be green or yellow. Shape can be constricted or inflated. Flower color can be purple or white. The size of the plant can be tall or dwarf, and the position of flowers can be axial or terminal. Chromosome numbers in cells. There are two types of cells that can be found in the body, the somatic cells and the sex cells. First, what is somatic cells? Somatic cells are the cells in the body of multicellular organisms that play no direct part in fertilization. Somatic cells undergo the process of mitosis for growth and development. In the process of mitosis, the body cells, also known as somatic cells, have the same number and identical chromosomes as the parent cell. They have two complete sets of chromosomes. Somatic cells are described as diploid because they have two sets of chromosomes. On the other hand, sex cells are directly involved in the fertilization. There are two types of sex cells, the egg cell and the sperm cell. A sex cell that undergo meiosis contains one set of chromosomes. These cells are called haploid. The matching pair in the diploid cells are called homologous. In humans, the diploid number of chromosomes is 46 and the haploid number of chromosomes is 23. Transfer of trait. Trait is different from gene, as traits is the distinguishing feature or character incorporated in a gene, while gene is a segment of DNA that codes for a particular trait and can be located in the gene. DNA is located in chromosome. The expression traits in each generation of offspring is controlled by the genes inherited by the organisms. It can be expressed into two, the genotype and the phenotype. All the genes present in the cells of an organism make up the genotype, while the phenotype is the physical expression of the genes, which is determined by the interaction of the genotype with the environment. So what we see in an individual literally is the phenotype, while genotype is what makes that appearance look like that. Non-Mendelian pattern of inheritance. Pattern of inheritance in which do not segregate in accordance with Mendel's law. Mendel's law includes law of dominance and uniformity, law of segregation of genes, and law of independent assortment. There are two types of non-Mendelian pattern of inheritance. First is the incomplete dominance and the codominance. So let's tackle about the incomplete dominance. The appearance of a certain trait can be a mixture of contrasting characters of a purebred parents, such that the combination of the two traits results to an intermediate expression of both traits, or a blended trait that does not resemble to the pure trait of either parent. For example, is like mixing a color. Let's have an analogy for this incomplete dominance. You have a two pure color, the red and the blue. When you mix those two colors, it will produce another color, which is the purple or violet. That violet resembles neither of the two colors, which is a new color or the intermediate color between the red and the blue. That is an example of incomplete dominance. Short activity. Problem. Coat color in mice is incompletely dominant. Yellow and white colored mice are homozygous, while cream colored mice are heterozygous. If two cream colored mice mate, what phenotypic ratio can we expect of their offspring? Show the Punnett square. Legend: YY for yellow colored mice, WW for white colored mice, and YW for cream colored mice. First, to solve this problem, we are going to draw Punnett square. Then we are going to put the allele of the parent. Since it says two cream colored mice, the allele that we are going to use is YW. So we are going to put YW, and to the side, another side, Y and W. To solve for the offspring of these two cream colored mice, we are going to multiply or cross the allele YY to have double Y, YW, and 2W. So what will be the phenotypic ratio of the offspring? Since in the Punnett square we see that there is one box that contains YY, therefore its percentage is 25% yellow according to the legend. And we have also two boxes with YW, therefore it will be 25% plus 25%, 50, and YW cream, so 50% cream colored mice. And the last one is one box that contains 2W, which means 25%, and 2Ws that stands for white, therefore it will be 25% white. So if you got the correct answer, then good job. So let's proceed to the next pattern of inheritance, the codominance. Codominance is another form of intermediate inheritance in which neither allele is recessive or masked, but the other allele in a pair that codes for any given trait. Both alleles are equally expressed and their features are both seen in the phenotype. Neither allele is recessive or masked in any of the generation of offspring in this pattern of inheritance. An example of codominance is the inheritance of type AB blood, wherein individual receives the type A allele from one parent and type B allele from other parent. The individual has both characteristics of type A and type B. There is no recessive in a codominance pattern of inheritance. In some traits, both alleles in the heterozygous are fully expressed. Codominant alleles are written as capitals with superscripts. Okay, let's try this problem. When a horse that is homozygous for red coat color is crossed with a horse that is homozygous for white color, the offspring would be what? Okay, again, we will use the Punnett square. So we are going to use C for the coat and the R and W, which stands for the color, R for red and W for the white. Okay, so cross each allele from the two horses. Okay, so we will have CWCR, 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 CWCR. So we have 4 CWCR, which means the genotype is 100% CWCR. For phenotype, since this is codominance, we will not be using pink since it is a combination of red and white, but we are going to use Roan, since in codominance there is no intermediate color that is produced between the parent. Therefore, the color or Roan, which means color of the same parents, are inherited but not mixed or fused. The result of the cross shows 100% Roan colored coat horse. Okay, let's try this. In a certain fish, blue scales capital B and red scales small b are codominant. What happens if you breed a patchwork of capital B small b fish with a fish that has only blue scales? Question: what is the probability of having a fish with red scale? And what is the probability of having a fish with patchwork scale? Can show them in a Punnett square. Then it says, what happens if you breed patchwork fish BB with the only blue scale? In this part, we will be using the patchwork fish BB, then blue scale fish BB, then cross the alleles. We have one box containing BB, 25% capital B, blue. So we have 25% blue. Then it shows 3 boxes filled with B and small b. Therefore, 25 plus 25 plus 25 is 75%. B. Question: what is the probability of having fish with red scale? The symbol for red scale is 2 small b. Since we have no BB or small bb in our Punnett square, therefore the probability of having a red scale fish is 0. For question number 2, what is the probability of having a fish with patchwork scale? Patchwork, capital B, small b, we have that in our Punnett square. We have 3. 1, we have 2, then the 3. Therefore, the probability of having fish with patchwork scale is 75%. Okay, so that ends our part 1. Now let's continue the part 2 of this video. Okay, so let's start the part 2 of this video. Multiple alleles. Genes could only have two alleles of contrasting traits such as tallness or shortness. But some genes can have more than two alleles, and these are called multiple alleles. If these genes undergo meiosis, an individual can carry two possible alleles, one for each chromosome. Take note that even if there are multiple alleles found in a particular gene, an individual can only have two of those alleles. Pair of any of these alleles can produce different phenotypes. ABO blood group in human is an example of multiple alleles. Blood types. Blood types are phenotypes that can be produced by three different alleles. For A, we have IA. For type B, we have IB. And O, we have small I. Now, this capital I means immunoglobulin, or the component that can be found in the blood type. Although there exist trait alleles for this trait, an individual receives two of these, one from each parent. Allele A and B are dominant while O is recessive. Since blood type is multi-allelic, its expression in the individual's phenotype is varied. A person can have one of four possible blood types: A, B, AB, and O. ABO, this is the chart of the ABO blood group. Okay, so in this part we have the father's blood type. As you can see, there are two letters because this is alleles. AO, that is blood type A. Then BO, blood type B. If OO, that's a blood type O, and AB. Then this second row shows the blood type of the mother. Now, this letter that you can see in your screen on a vertical way is the possible blood type of children or offspring. If the father and the mother has the same blood type, which is AO, possible blood type for child would be A and O. If parent has type AO and BO, possible blood type of their offspring is one of the four. Can be A, B, O, or AB. If the parent is AO while the mother is AB, there are three possible blood types: the A, B, and AB. For AO and OO, there are only two. Possible blood type would be A and the O. For BO and AB, possible blood type of children would be A, B, and AB. For OO and BO, possible blood type would be B and O. For OO and AB, blood type would be A or B. If father and the mother has the same blood type OO, possible blood type of children is O. While for AB and O, possible blood type would be A and B. While try this, Derek is homozygous for blood type B allele, while Nikki is type O. What are possible blood types of their baby? Since parents are both homozygous, therefore their blood type would be, for Drake, B, and for Nikki would be O, O. If you cross, multiply, BO, PO, PO, PO. So what are the possible blood types of their baby? Since in the box of the Punnett square that we have computed shows BO at 4 squares, which are equivalent to 100% since each BACA is 25%. Possible blood type would be B. So the combination of the blood from Drake, which is B, and Nikki, which is O, the blood type of their baby would be type B. Next, polygenic traits. Polygenic traits are phenotypes that are controlled by more than just a pair of genes. For example, human eye color results from different gene pairs that accounts for the tone and pigment distribution. Multiple alleles should not be confused with polygenic traits. In multiple alleles, there are many different alleles from a gene. In a polygenic traits, an individual has many different gene pairs working together to produce one trait with additive character. That ends our discussion today. I hope you learned new knowledge from me and you enjoy watching this episode.